My name is Luke Gilbert. I'm an assistant professor in the Department of Urology in the Helen Diller Family Comprehensive Cancer Center, as well as the Innovative Genomics Institute of UC Berkeley and UCSF. I'm a synthetic biologist who seeks to apply new tools to understanding human disease. We come to this from the perspective that the human body is encoded by at least 200 cell types, each of which has the exact same genome, but these cell types have widely different functions if you think of a neuron versus a muscle cell. So it's not the genome that specifies cell function, but instead the coordinated expression of the genes that are transcribed in each cell type. And so we are seeking to use new tools to understand this complex problem. And what we've done is over the last several years build a toolbox of repurposed CRISPR epigenetic editors that allow us to edit both the histone-based protein epigenetic code as well as the DNA methylation code, and turn genes on or off at will in a very precise and specific way. And so you can think of this as turning genes on or off as a light switch, but we can also turn things up or down in a programmable way, more like a rheostat. So we're manipulating gene expression the way electricians might manipulate electricity in a wiring diagram. And so our goal is to turn genes on or off and ask what a cell needs to function normally and when it needs it and why it needs it. This in and of itself is powerful, but what we've done over the last couple of years is also build a platform in which in a single experiment we can turn on or off all of the genes that are encoded by the human genome one at a time in each cell, but in a pooled fashion. So we can take a graduate student and in 10 weeks ask how every gene encoded by the human genome is important for some specific cellular process. This, though, represents a bit of an oversimplification in how cells actually work. And so we know that for many cell properties, it's not a single gene that's important, but the coordinated regulation of multiple genes in governing cell properties. And so we've also, over the last couple of years, built a platform in which we can turn on or off pairs of genes. This enables us to map how genes work together to control cellular properties. Another term for this is a genetic interaction or an epistasis. And in manipulating two genes is valuable, but same as before, what we really want to do is manipulate large numbers of pairs of genes. And so in our initial experiments, we manipulated about 250,000 gene pairs, enabling us to, in a high throughput quantitative way, map how genes work together to control cell physiology. Our goal is to further scale this up, and so we have a plan to construct an experiment in which we manipulate 10 million pairs, and what we're going after is the genes whose proteins are localized to the nucleus, the human nucleus. And so what we'd like to do is create an unprecedented high-resolution map of genes that are proteins that are localized to the nucleus, enabling us to understand how the genome is controlled and how the output of the genome is controlled more specifically, because we're interested in transcription and how gene expression controls cell identity. And so we hope this will be both a resource for the community in creating a very high-resolution map of genes that control nuclear activity and also enable insights into human disease. My lab is also heavily invested into prostate cancer and lung cancer research. We're using all of the tools that I described to go after pressing problems like metastasis and drug resistance. Lastly, what we're really excited about recently is turning these research-grade tools that I've described into an actual therapeutic. And so we think we've come up with ways where we can have a transient delivery of our epigenetic editors and seed a stable form of epigenetic editing that can last for months or years. And so we're putting this into animals right now, and we would hope in the next 5 to 10 years, if this is successful, to go into humans as a new form of medicine, where instead of using small molecules or protein-based therapeutics, we're instead just using epigenetic medicine to turn genes on or off directly. And the first application we're using is as an antiviral, where we're just turning off the viral receptor, the virus can't invade anymore, and this represents, in essence, a new immunization strategy. And so we're delighted to be collaborating heavily at UCSF, and we're excited about the next couple of years.